Hello, everyone, and welcome to Humanist Canada's monthly webinar series. I am Anna Popovich, Program Director at Humanist Canada, and I will moderate today's conversation. As always, this webinar will be 60 minutes in length with a Q&A period at the end of the presentation. If you have a question and would like to ask it directly, please raise your hand at the bottom of the screen, or you can submit your question or comment with the Q&A tool that you can also find at the bottom of the screen. This webinar will be recorded and uploaded to our website. Our topic today is All in the Human Family, Neanderthals, Denisovans, and Us. And our guest today is Dr. Bence Viola, a paleoanthropologist specializing in the biological and cultural interactions between different hominin groups in the late Pleistocene. Hope I'm pronouncing this correctly. After studying at the universities of Vienna and Bordeaux, he spent 4 years as a postdoctoral fellow at the Max Planck Institute of Evolutionary Anthropology in Leipzig, and is now an associate professor at the Department of Anthropology at the University of Toronto. Dr. Viola's research uses an interdisciplinary approach, combining morphological, archaeological, and genetic data to better understand how the Neanderthals, their enigmatic Asian cousins, the Denisovans, and the first modern humans interacted. He has led excavation in Central Europe, East Africa, and Central Asia, and is currently conducting fieldwork in Kyrgyzstan and Austria. Welcome, Bence. Great to have you, and thanks so much for joining us. Over to you.
Thank you very much, Anna, and thank you very much for attending. Let me just share my screen, and then we can get started. There you go. I hope everyone can see this now.
Yep.
So today I would like to talk a little bit about how our understanding of human evolution was transformed in the last about 15 years by research using new molecular techniques that allows us to have a much more nuanced understanding of the interactions between different human species. So today, as I think everyone knows, humanity is a single species. We are all modern humans, what we are Homo sapien, what we call Homo sapiens scientifically. In the past, though, this is a little bit different, and this is actually a trope that is quite frequent in fantasy and science fiction. You know, in the Lord of the Rings, you have the different kinds, the different species, the elves, the dwarfs, the humans, the hobbits, all living together. Similarly, in science fiction, you have the same thing. The fascinating thing is, though, that this was actually true for a large part of our species' life. It's only the last about, let's say, probably 30,000 years that we are alone on this planet. For a long time, human evolution was seen as something very linear, from a chimpanzee-like ancestor towards us, through thought to go through very gradual steps, but mostly in a single direction. Today, we realize that it's much more complicated than this. You have lots of different species that more or less tried out different adaptations that went towards humanity. It's possible that even bipedalism, upright walking, arose independently several times among some of our ancestors. And what we also understand now is that these lineages were not completely independent. Whenever they met, they actually exchanged genes. And today, we are kind of a mixture of lots of different ancestors. So this is just one view of the last about 1 million years. And as you can see here, if you just look around, let's say about 100,000 years ago, or between 50 and 100,000 years ago, you see that, so time is on the y-axis, and on the x-axis, you have different species or populations is probably the better way to call it. And as you can see, between 50 and 100,000 years ago, you have what is described here as later or modern Homo sapiens, which are direct ancestors. But you also have late Neanderthals, Denisovans, which is a group that I'll talk about extensively today, potentially another group in China that probably goes extinct around this time, Homo erectus in Indonesia, Homo floresiensis, the hobbits, which is this dwarf hominin known from the island of Flores, and then a species about which we know practically nothing up until now that has only been discovered in 2019 in the Philippines, Homo luzonensis, which also seems to be a very, very small hominin. So, this is clearly a much more complicated picture than a single linear lineage evolving towards us. Today, I'll be focusing on the geographic area where I've worked the most, simply because this is the area I'm the most familiar with. And luckily, this is also an area that over the last 10 years or 15 years turned out to be one of the most important areas for better understanding our evolution, because here in Central and Northern Asia, we actually have 3 different hominin lineages that co-occur: Neanderthals, Denisovans, and our direct ancestors, modern humans. And so we can look at their interactions in quite a bit of detail. So, let's talk about Central Asia in kind of the wider sense, as I would like to call it. This is a very interesting region because it's a region of extremes in many ways. You have some of the driest deserts here, the Taklamakan and the Gobi. You have some of the highest mountain ranges in the world here with the Tian Shan and Pamir, different parts of the Himalayas, but you also have more medium mountain ranges such as the Altai and Sayan mountains. And then if you go towards the west, you actually are connected to the Great Eurasian Plain, the steppe belt that stretches from Hungary into China, and that probably played an important role for migrations, because once you were adapted to hunting on this steppe belt, as Neanderthals, for example, were, you could probably migrate relatively easily east-west. The more complicated issue is that probably from time to time, parts of this steppe belt were cut off by transgressions of the Caspian Sea, which during cold periods had a much higher sea level than today by about 120 meters. And that meant that it extended about 1,000 kilometers further north than it does today. And also by huge lakes and swamps in Siberia. But in general, this was probably an area that was easy to traverse. To the north, the region is delimited by the boreal forest. Again, an area about which we know remarkably little, simply because organic preservation in these areas probably is in general bad. Bones decompose. And also today, it's a relatively hostile area, and so there's much less occupation than elsewhere. So there's no construction that would lead to the discovery of archaeological sites and so on. There's also not many caves where we could look. So we know very, very little about this. We know that some people were up there. There's been recently discoveries even up by the Arctic Ocean. There's a paper that claims that Neanderthals might have reached the Arctic Ocean towards the north end of the Urals. I'm not completely convinced, but it's pretty clear that some people were up there by about 45,000 years ago. Research in this area is not something completely new. Roy Chapman Andrews, who was a paleontologist at the American Museum of Natural History, started several expeditions in the 1920s, the so-called Central Asiatic expeditions. Roy Chapman Andrews is a very colorful person. It is said that he served as kind of the inspiration for Indiana Jones. So in one of these expeditions, we know, for example, that they started out with 3 cars, but also 200 camels that carried the gas for the 3 cars. Chapman Andrews hoped to discover some evidence for the earliest humans in Central Asia, and what we called Inner Mongolia at the time. He was not successful in this regard, but on the other hand, he was actually very successful from a paleontological perspective. He discovered the rich dinosaur fossil fields of the Gobi. He was the first one to discover fossil nests of dinosaurs and similar things. Research in the region then stopped quite a bit and only restarted quite intensively in the 1980s by Russian scholars, and then more recently by people cooperating with them. I want to take you first to one area that is probably the area that lies the closest to my heart, the Altai Mountains of Siberia, and in particular one site that is really the key site for our understanding of what's been going on in this region, and that is Denisova Cave in the valley of the Anui River in the foothills of the Altai Mountains. Now, this is southern Siberia. So, despite people frequently imagining Siberia as a frozen wasteland, it's pretty clear that this is not what we're looking at here. This is a medium mountain range. The Denisova Cave is roughly at the same latitude as Leipzig in Germany, so further south than Berlin or Amsterdam or similar things. So it's not Arctic at all. This is the view from above the cave that I think a little bit shows also why the people might have inhabited this area. These valleys through the foothills likely served as highways for migrating herds of horses and reindeer. Then up in the hills, you had things like ibex, and depending on the climate, maybe roe deer and red deer. Even mammoth and woolly rhino came through from time to time. And so the people sat by these caves. I'm standing here right above the cave, looking down into the valley. The people sat in front of these caves and waited for animals to come by and then hunted them. And what you see down there in the valley is actually our little scientific camp. My colleagues from the Russian Academy of Sciences built since the 1980s a really nice camp with housing for visiting researchers, for students, a communal kitchen, labs, storage areas, and so on. It's actually very pleasant to work there. This is the entrance of Denisova Cave. The Denisova Cave was named after a hermit, Denis, who lived there in the 18th century, supposedly. As you can see, you have a larger entrance on the left and then a small entrance on the right. The large entrance on the left leads you into the main chamber, and then on the right, we get into the so-called South Gallery. Caves are always relatively complex, and so, I can't go into too much detail. Caves are complex environments. They are inhabited by lots of different animals. In the case of the Denisova Cave, for example, humans were probably not the main inhabitants. The main inhabitants were actually cave bears, hyenas, even cave lions. And so lots of the bones that we find in there were brought in by these carnivores. Humans were likely only temporary visitors from time to time. Even more, of course, you had bats in there. You see the excavations here a little bit. This is, I think, in 2018. And you can see on the top of the sequence, you see this very finely laminated kind of striped horizons. Those are Holocene. Those are from the last 10,000 years. And they're actually deposits of guano mostly and ash. So the cave was used by the local herders as a stable for their sheep and goats mostly. And what they learned is that from time to time, you have to burn the accumulated goat and sheep crap. And so you get these very finely laminated ashy deposits. But underneath, in the area where the students are kneeling, we are actually in the Pleistocene. These are deposits that go from about 12,000 years back to potentially about 300,000 years. And these contain evidence for visits of humans, but also of lots and lots of animals. The stratigraphy is very complex because in caves you have lots of different processes. You have, in this case, for example, you have a chimney in the main chamber through which water was flowing in from time to time that moved around the sediment. You also have, of course, rock falling off from the roof and from the walls. You have the various creatures, in particular the hyenas, digging around. So it's actually quite hard to figure out how the different parts of the cave are related to each other chronologically, which deposits from which part of the cave are contemporaneous. And there's lots and lots published on this. In the case of Denisova, if you're interested, there's a paper by Jacobs and colleagues from 2019 that probably has the most details, but I think this is enough. For me, the Denisova Cave is mostly interesting because it also contains some human remains. And the most famous of them is this little piece here, Denisova 3, which looks completely unspectacular. A tiny piece of bone here on the finger of my colleague, Susanna Sawyer. This is part of the finger bone of a young girl. We guess she was somewhere between 14 and 16 years old at the time that she died, or at the time at least that she lost her finger. As you can imagine, finding such a finger bone is relatively hard. In this case, it was found when my Russian colleagues were sieving for microfauna, looking for tiny little bones and teeth of rodents, bats, and similar things, which are very useful for reconstructing the environment at the time of the inhabitation of the cave, and also help us to date it a little bit. So while looking for these rodent teeth, they found a tiny piece of bone that they actually realized was part of a finger bone, and they sent it to the Max Planck Institute of Evolutionary Anthropology in Leipzig, where I worked at the time, to analyze. Now, I was familiar with Denisova Cave already because I studied 2 teeth from there for my PhD, and I knew that, you know, based on the date of the deposits, the most likely explanation for the specimen would be that it would be a Neanderthal. But as you can see, as you will see, this was not really the case. So this is a close-up of micro-CT scans, so very high resolution computed tomography scans of this bone. And as you can see, it actually consists of 2 pieces. The blue part is the articular surface that's between the last phalanx of your pinky, the last segment of your pinky, and the middle segment. And the green part is part of the shaft that then goes up to the tip of the finger. And as you can see, these are not completely fused, which shows us that this is actually an individual that is still growing. But towards the bottom, we can actually see some evidence for the fusing having started, and that's why we think it's a 14 to 16-year-old. That's when it happens in people normally. Now, this was not the only fossil from the cave. We also had a tooth that was found a little bit earlier, but our Russian colleagues only found it. It's a bit of a tragic story. The day that this tooth was found in 2002, a student excavating at the cave was killed by a truck going by on the road. And so they completely forgot about this tooth, obviously. And only about 10 years later, in 2010, my colleague Anatoly Derevyanko, he pulled it out of his shirt pocket and showed it to me. And looking at this tooth, it was very clear to me that this is something very special. This is an absolutely humongous tooth with gigantic roots. But again, you know, it's just one tooth. So what can we tell from that? One method that really revolutionized our understanding of human evolution in the last years is the use of ancient DNA, using DNA from fossils to better understand both their phylogenetic position, but also a lot of other things that I'll talk about. So I'll give you just a very short overview of how we do this. Of course, in reality, this is a very, very technical approach, but I think we can just look at it for a few minutes. So in the end, what you have to do is you have to get some bone powder, which sadly is destructive. So we are usually quite careful with it. This is from a very early study where they actually removed about 5 grams of bone. Luckily, today we're down to maybe 20 to 30 milligrams, so we don't have to cut out huge pieces. Still, it's a destructive analysis, and so many curators are very cautious in when we do whether we can do this and whether it's really worth it. Once you have some bone powder, we use various enzymes that digest things like the proteins and that remove the mineral matrix of the bone. And we more or less purify it so we are left just with the DNA in there. And then we amplify the DNA, meaning we make many, many copies of it. And then we use different methods to sequence it, to read out the sequence of the bases that write out the DNA. Now, again, you know, this sounded very quick. In reality, this is something that involves lots and lots of technology, lots and lots of know-how, and also lots and lots of money. This is not cheap. Now, I'm sure you remember from Jurassic Park that, you know, people can extract dinosaur DNA. Of course, in Jurassic Park, this was science fiction. The amazing thing is that actually in the last about 15 years, this became reality. So if you look at recent DNA, we have about 1 microgram of DNA per gram of tissue, which, you know, is quite a bit. The problem is that over time, DNA decomposes, and we have much less of it. Another problem is the issue of contamination. You know, DNA, because such very small amounts are present in tissue, you know, it's very easy to get some contamination in there, even smaller amounts. The problem with ancient DNA is that there is much less of it than recent DNA, by about 1,000,000-fold in some cases. And not just that there's much less DNA left, it's also not in long strands anymore. You know, in our cells, DNA is more or less one continuous strand of the whole genome, about 3 billion base pairs in the case of the human genome. In the case of ancient DNA, various processes, chemical degradation, but also the activity of various bacteria and so on, breaks down the bacteria into little pieces. So instead of a strand that's millions or billions of base pairs long, we actually have pieces that are maybe 30, 50, 100 base pairs long. Just imagine you take a whole library and you put it through a shredder, and then you throw away 99.9% of it, and then you just have those little pieces that came out of the shredder, and you try to reconstruct the library. It's very difficult, but surprisingly, it's actually possible in some cases, simply because, to continue with the library analogy, maybe you have many copies of certain books in the library, and if you have many copies, then potentially enough parts survive that you can piece those together. So besides the DNA being fragmented and being very little, it also has very characteristic damage patterns, usually towards the ends, that seems to be a problem, but actually in reality, we use this to authenticate ancient DNA. So if we see these characteristic damage patterns, we know that it's actually old DNA and not DNA from, whoever excavated it or so. An even bigger problem is that we have a lot of what we call non-endogenous DNA from bacteria and fungi and, the cave hyena that potentially chewed on the bone and so on. Which we also have to separate. But again, because you can use these pieces of DNA and you look them up in a database, and you can see whether this fragment, for example, shows up in the human genome, or it shows up in the hyena genome, or in the genome of some fungus. And if it's for some fungus or the hyena, you throw it away and you keep it if it's part of the human genome. Contamination is an even bigger problem with these old specimens, simply because there's already much less DNA, and so any contamination makes up a much larger proportion. Despite all these problems, surprisingly, this actually works. We can get DNA out of these specimens, and we can actually get very interesting results from this. So, for example, the first analyses that we did, we looked at the mitochondrial DNA of this finger bone from Denisova Cave. Mitochondrial DNA is a small, kind of ring-shaped DNA molecule, a bit like a bacterial chromosome, that is part of our mitochondria, organelles of the cells. And it's only about 17,000, 18,000 base pairs, depending on what species you're looking at. So much less than the 3 billion base pairs of the human genome, and thus much easier to assemble, also because there are thousands of copies of mitochondrial DNA in each cell, while only a single copy of the nuclear genome. So when we looked at the DNA of this finger bone from Denisova Cave and used the molecular clock, which is a method that allows us to tell when the last common ancestor of 2 DNA sequences lived, we saw that this finger bone was not a modern human, but also that it was not a Neanderthal. It was something completely different. It was something that separated from us modern humans about twice as much time ago, about 1 million years ago, than Neanderthals. Neanderthals separated from us based on mitochondrial DNA about 500,000 years ago. The separation from chimpanzees is somewhere between 7 and 11 million years ago. Again, there's a lot of assumptions in these numbers. The exact numbers actually vary a lot depending on what kind of analyses you do. But what matters is the pattern, is that Neanderthals and modern humans are more similar in their mitochondrial DNA, and this finger bone from Denisova Cave is something different. So based on this, we propose that this is a population called the Denisovans, just like the Neanderthals. There's one problem with this, though. Mitochondrial DNA is easy to analyze, but it has one very unpleasant property, and that is that it's matrilinear. We inherit our mitochondrial DNA from our mother, and she inherited it from her mother, and she inherited it from her mother, meaning that as we go back in time, if we look at mitochondrial DNA, we're only looking at a very limited portion of our ancestry. We're not looking at all of our ancestors. We're only looking at one of them in each generation. And this can create a lot of problems. One of them is that mitochondrial DNA can actually be misleading in such phylogenetic analyses. And I think the easiest analogy for this is if you think about surnames. You know, in Canada at least, and in most Western societies, traditionally, people inherit their surname from their father. So a father who only has daughters looks as if he would not have any descendants if you just go by the names. And so surnames can, get lost very easily, even though these individuals have descendants. Same thing can happen with mitochondrial DNA. If a mother only has sons, if a mother has a son, that son carries her mitochondrial DNA, but the grandchildren don't carry the mother's mitochondrial DNA anymore. They carry the mitochondrial DNA of the maternal grandmother. So this can lead to weird effects and can be misleading. And when we analyzed the nuclear DNA of this finger bone from Denisova Cave, we actually got a very different result. We actually found that this finger bone and this population in Denisova Cave is more closely related to Neanderthals than they are to us. We found that the common ancestor of Neanderthals and the Denisovans lived maybe 450,000, 500,000 years ago, while the common ancestor with us lived about 650,000 years ago. Either way, it was clear we have something completely new here, a population that was previously unknown. And when we published this in 2010, this was quite a big splash. It was on the title page of the New York Times, which is, not that frequent in science that you find your research there. It was quite neat. Because we had the complete nuclear genomes, we actually could do much more refined analyses. We could look at how these populations are related, both Neanderthals and Denisovans. We had a Neanderthal genome from Europe that we got around the same time. We could look at how they are related to various present-day populations. So we picked 5 modern humans, or I'm just using 5 here for an example. In reality, we've done this with thousands. But if you pick 5 modern human genomes, one, a Khoisan from South Africa, a Yoruba from West Africa, a European, which in this case was a French, a Han Chinese, and a Papuan from Papua New Guinea. And we compare them to these archaic genomes. The best way to do this is using something we call Abba-Baba or D-statistic. And I'll explain this shortly because it's remarkably simple and elegant, and I'm always astonished that something so simple can actually work. So what we do is we compare the genomes of 2 recent humans. Let's say in this case, a French and a Yoruba, so European and West African. And we find positions in their genome where they're different, where there's a difference between these 2 individuals. And then we check in our archaic human, whether in a Neanderthal or in a Denisovan, whether they have the same variant as the French individual or the same variant as the Yoruba individual. And theoretically, if the archaic human is as closely related to the French as to the Yoruba, you'd expect that in 50% of the cases, you have the same variant as the French, and in 50% of the cases, you see the same variant as the Yoruba. Now, we do this with like 25 million variants in the human genome. And when you do it with that many, you realize that the proportion is not exactly 50-50. There's a slight bias. And what you find with Neanderthals is that in this case, they are a little bit more similar to the French. You'd find that maybe in 51-52% of the cases, they are similar to the French, while in only 48-49% of the cases, they are similar to the Yoruba. So I just drew in here in red kind of the way we imagine that modern humans migrated out of Africa. So our direct ancestors likely all originated in Africa in the last about 100,000 years. And then we moved out of Africa. And so what we think what happened is that as we moved out of Africa, we met some of these archaic populations, Neanderthals and Denisovans, and we interbred with them. So if you look in African populations carry 0% of Neanderthal DNA, and that's why they are a little bit less similar to them than other populations. Europeans, Asians, and Melanesians all carry about 1 to 3% more Neanderthal DNA than African populations. which indicates that as we came out of Africa, we met the Neanderthals, we interbred with them, and we picked up a few variants of Neanderthal DNA. We can do the same analyses with the Denisovans as well. And if we do that, we find again, there's 0% Denisovan DNA in Africa, there's 0% in Europe. Now, my expectation was if we find somewhere any kind of evidence for modern humans mixing with Denisovans, at the time we only knew Denisovans from Denisova Cave in Siberia, I expected, that would be somewhere in Asia. And we actually found about 0.1% of Denisovan DNA in present-day Asian populations. What was remarkable, though, is that as you get down to Melanesia, you actually see about 4 to 6% of their DNA comes from Denisovans, in addition to the Neanderthal contribution, which, to me said in particular one thing: Denisovans must have been much more widespread, because the ancestors of present-day Papuans definitely did migrate through Siberia. That would make no sense. So with these genetic analyses, we found a pattern of multiple hominins that were in contact. Whenever these populations met, they interbred with each other. And we see this now everywhere. We even see gene flow between Neanderthals and Denisovans, and I'll show you an example later. There's gene flow between Neanderthals and modern humans, and there's even gene flow from what we call a potential unknown hominin into Denisovans. That's a more complicated story. So what was interesting, though, around the same time that we found and got these results, is that we found that in Denisova Cave, we don't not only have Denisovans, but we also have Neanderthals, because we found this toe bone, Denisova 5, that turned out to be a Neanderthal based on its mitochondrial, but also based on its nuclear DNA. And so one of the neat things that we could do with this individual, because the preservation of the DNA was so good, was that we could actually look at its genome in more detail, because our genome in the end samples 2 different individuals. We have 2 copies of each chromosome, one that comes from our mom and one that comes from our dad. And actually, if we have enough DNA from a fossil, we can actually compare the chromosomes that came from their mom and the chromosome that came from their dad. And so this allows us to tell a little bit about how closely the parents were related, and based on this also how large the population from which they came from is. And so one of the ways to plot this is these kind of analyses. So what you have here, it looks a bit like an ECG, is actually one chromosome. This is chromosome 1. And what you see here is when the red line is close to the bottom of the plot, that means that the heterozygosity, so the difference between the 2 chromosomes that we got from the mom and from the dad, is very low. And where it goes up, where you have a peak, that means those are areas where the heterozygosity is high. So those are areas where the 2 parents were very different. And as you can see in the French, you have quite a bit of difference between the parents. Denisova is the Denisovan, and there already you see that in some parts we actually have less variability than in present-day humans. What is striking, though, is the last one, Altai, which is this toe bone from Denisova Cave, the Altai Neanderthal, where you have whole pieces of the chromosome. There's like a segment that's almost half the chromosome where the 2 parents were identical, indicating that we're actually very closely related. And what we see in this individual actually is extreme inbreeding, a bit like a European royal family, like the Habsburgs or so. So the parents of this individual were so closely related that they were either grandfather and granddaughter, they could have been half siblings, they could have been uncle and niece, or double first cousins. Now, this is kind of gross, but understandable. They were likely living in very small groups, and how small, we'll see that in a moment. What is interesting is we can actually remove the effect of this inbreeding using various statistical methods. And even if we remove this last generation inbreeding, it looks as if this population was extremely small, smaller than any present-day population of modern humans. Even, the most isolated indigenous tribe in the Amazon has more genetic diversity and likely a larger population size than these Neanderthals in the Altai. So how did Neanderthals get into the Altai? Neanderthals are a species that is usually assumed to be primarily European. Like the fossil record of Neanderthals can be followed back in Europe to about 500,000 years. We see a slow accumulation of Neanderthal characters in Europe, starting with some sites in France, such as Atapuerca Cima de los Huesos, and then going through various other things like Steinheim and so on, Arago, until we have like fully Neanderthal-like Neanderthals by about 200,000 years ago there. So these Neanderthals somehow migrated into the Altai. And what is fascinating is that this likely happened more than once. And I'll show you the evidence in a moment. One of the cool things is that we can actually use these genomes to reconstruct population size over time. And this works by, taking a genome and chopping it up into little pieces. And for each of these pieces, looking when the last common ancestor of this genome and lots of other modern genomes lived. And using this, we can statistically reconstruct population size. I mean, theoretically, genetic diversity, but it's a good proxy for population size. And what we can see here, so here on the right, on the y-axis, you have time. As you can see on the top, it goes back to somewhere between 5 and 10 million years ago. On the y-axis, you have population size. It's effective population size, but it's okay. And as you can see, is that between 5 to 10 million years and roughly somewhere between 500,000 and a million years ago, Neanderthals in red, Denisovans in black, and then all the other colors are various modern human populations. They all were a relatively large population that then became a little bit smaller, lost genetic diversity. And then Neanderthals and Denisovans separate. They start losing genetic diversity very quickly. There's less and less of them. And this is probably the time when they left Africa, about 500,000 years ago, maybe, and moved Neanderthals into Europe and Denisovans into Asia. Modern humans in Africa, in the meantime, increased their population size. But then you also see another huge drop starting at about 150,000, 100,000 years ago, where the green and yellow lines especially go down. And this is what we call the out of Africa bottleneck. This is a strong loss of genetic diversity at the time of the ancestors of most modern humans leaving Africa. And then you see it's only the last about 10,000 years that population size explodes, in particular for the Han Chinese, where, over the last few thousand years, you went up to now about 1 billion people. Neanderthals and Denisovans never went through these increases in population size. They had smaller and smaller and smaller populations until they slowly disappeared and died out. So I want to talk shortly about another cave too near Denisova that we've been excavating since 2008, Chagirskaya Cave, which is amazing because we only have Neanderthals there. But it's nice for me because we have lots and lots of them. We actually have the remains of probably about 15 individuals. We have, for example, one individual for which we have most of the arm. We have lots and lots of isolated teeth. We have pieces of a foot. We have some vertebrae. We have a nice mandible. And what is nice about these is that we can actually tell morphologically without the DNA that they are Neanderthals, because they are very similar to what we see in Europe, for example. This mandible is a very characteristic piece. For example, it has this oblique ridge that's descending here from the top right towards the bottom of the bone. Somehow I can't show something. I can't point at stuff at the moment, but I think you can see it. There's also some traits on the teeth. We have little crests that I marked here in red that connect the two mesial cusps, the two cusps in the front. Again, something we see in Neanderthals, but don't see it in any other species. So, this is clearly a Neanderthal. Also, the teeth we have, for example, on this incisor, this is what we call incisor shoveling. Again, a typical Neanderthal trait. And so the nice thing is we had all these morphology, which told me that they are Neanderthals, but then we also got some DNA out of this group, including, in part from this one finger bone. And the amazing thing, what we found is that even though Chagirskaya Cave is only about 100 kilometers from Denisova, this is not the same Neanderthal population. This is actually a different Neanderthal population, much more closely related to European Neanderthals, which indicates that this migration from Europe into the Altai didn't just happen once, it actually happened twice. And you can see this based on the phylogenetic tree down here at the bottom. And what is even nicer is that last year we managed to actually extract DNA from several different individuals. And we could show that the individuals whose bones we find in this cave are very closely related. We actually found a father and a daughter in there. We found a second-degree relationship, which is something like a grandparent and grandchild, or potentially cousins. But even all the other ones seem to be very closely related. And again, we were able to look at the genetic diversity in this population and the population size, and we found, even though this was a second independent Neanderthal population, it was just as small as the other one. And this to me means that these really were Neanderthals at the very periphery of the Neanderthal range. They were isolated from other groups. We tried to simulate how much gene flow there could have been, and it's really minimal. If we assume a group size of maybe 20, 30 individuals, it's every 10 generations that they'd get another individual in from a bit further away. Which is not enough and probably not healthy, and might have been part of the reason why they disappeared. These small population sizes lead to what we call allele effects in ecology, when you just don't have enough partners for reproducing. And of course, the inbreeding can lead to various health issues as well. Actually, the genetic diversity we see in Chagirskaya is comparable to what we see in endangered species today. For example, in mountain gorillas and eastern lowland gorillas. So, you know, these Neanderthals were really on the brink of extinction at the time. One of the fascinating things that we also found is that we actually saw more diversity in the mitochondrial DNA than in the Y chromosome. The mitochondrial DNA is inherited maternally, while the Y chromosome is inherited paternally. So this means that the women in the site were less closely related than the men, indicating that if their people came in from the outside, it was likely the women. So the men probably stayed with the group they were born in, but sometimes women came in from the outside. So a kind of a patrilocal society, which again, you know, it's kind of unbelievable that we can tell this 70,000 years after these people lived. Now, to kind of close up the whole thing, I wanted to talk shortly about some more about these contacts between hominin species, because we actually have direct evidence for this in the Altai. A few years ago, we found this tiny piece of bone, which looks really even worse than the phalanx. It's such a small piece that we could only identify it using molecular methods. We use a different method also based on proteins to identify bones called zoo archaeology using mass spectroscopy. And that pegged this bone as potentially hominin. And so we extracted DNA from it. And I mean, this fragment, based on everything else, I can't even tell which bone of a human it comes from. It comes from one of the large long bones, tibia, femur, humerus. It's probably digested by a hyena. The surface was quite eroded, but beyond that, we really can't tell anything. What was amazing, though, is when we looked at its DNA, we found that this is actually a Neanderthal Denisovan hybrid. This individual carries about half of its DNA is Neanderthal-like, and the other half is Denisovan-like. And we also know the mitochondrial DNA is Neanderthal-like. So the mother was a Neanderthal, while the dad was a Denisovan, which, you know, indicates clearly that these populations met in the Altai at the time of this individual's life, which was really pretty unbelievable when we found this out. And we could actually, based on a more detailed analysis of the genome, show that the father, despite being a Denisovan, actually already had some parts of the genome that were Neanderthal-like, indicating that there were already contacts between Neanderthals and Denisovans about 100 to 150 generations before the father was born. So we see repeated contacts between these populations. So you see here the common ancestor of Neanderthals and Denisovans in yellow around 450 to 400,000 years ago. Two populations separate. with the Neanderthals, again, splitting up maybe 150,000 years ago into the Altai Neanderthal, the Neanderthal from Denisova, and all the other Neanderthals from Europe and from Chigirska that we have, which is the blue here at about 100,000. Chigirska would be part of that. And then you have these two populations meeting as evidenced by Denisova 11, this hybrid bone, Denny. But, these contacts didn't just happen between extinct species. They also happened with our species. And the fascinating thing is that these actually mattered. There are some features in recent humans that can be explained or that are linked to the gene flow from archaic populations. One of the more interesting ones is that in our immune system, in the so-called HLA system, the human leukocyte antigen system, there are variants that came from Neanderthals and Denisovans. And so the HLA system encodes various receptors on white blood cells that are used to, capture little pathogens that come into us. And so the idea is that what you usually have in the HLA system is what we call balancing selection. So you want to have as much diversity in the population as possible, so you can catch as many different pathogens as possible. Now, when you go through a bottleneck, such as modern humans leaving Africa, you lose a lot of genetic diversity. And so interbreeding with Neanderthals and Denisovans allowed us to pick up some variants from them that increase the genetic variability, but also because Neanderthals and Denisovans were around for such a long time, they probably adapted to their environment and adapted to the pathogens that were there. And so we could pick up these variants that were, useful against pathogens present in Europe and in Asia, and use them. The other thing that's even more interesting is that Tibetans carry a certain variant in the so-called EPAS1 gene. EPAS1 is a hypoxia pathway gene. So a gene that actually, among other things, is linked to blood flow in the placenta during pregnancy. And we know that Tibetan women who carry this specific variant have larger babies, give birth to larger babies, which is important because birth weight is a very important predictor of infant survival, especially, without medical care. And this has been known since the 1980s, and it always looked like there's been very, very strong selection in a few of these genes in Tibet. And the fascinating thing is that Emilia Huerta Sanchez and colleagues in 2014 could show that the variant we see in Tibet actually is a variant that came from Denisovans. So Tibetans in general carry only about 0.1% Denisovan DNA, like everyone else in Asia. And 0.1% Denisovan DNA means across your whole genome, about 0.1% is Denisovan. And that is kind of randomly distributed, and everyone has different parts. But in this one part, in this one gene, in EPAS1, 80% of Tibetans carry this variant. So it's been selected for extremely strongly, because it allows you to survive at high altitudes and have healthy babies, which is pretty unbelievable. So I hope that I gave you at least a short overview of how our evolution kind of looked, how it changed over the last years, to this picture of many different populations that interact repeatedly. And I think at the end, I hope I could also show a little bit why this matters for us today, that there are actually features linked to our health and other adaptations that actually come from these archaic populations. There's, for example, also, I didn't show it, there's several genes that are linked to skin pigmentation. Light skin color, both in Europeans and in Asian populations, actually comes from Neanderthals, but they're different genes. So clearly, these interactions with other hominins were important. This research is research that happened in collaboration with many people, and I could have never done it alone. And I'm very grateful both for my colleagues in Leipzig and Vienna and the US for cooperating with me. but especially for my colleagues in Russia, Uzbekistan, Kyrgyzstan, and elsewhere, for their work at the excavations and their important discoveries, and also, of course, letting us work on this material. And of course, I want to thank you for your attention.
Thank you very much, Bence, for this fascinating presentation. Very cool. Please ask away your questions. I see we already have 4 questions in the Q&A. You can also raise your hand and ask it directly. I have a question for you. So are there any clues as to what the interaction between these different hominin groups look like? How did they live? Hey, would you like to mate with me? And who are you? You know, that sort of thing. How much do we know about that?
I mean, you know, that is, of course, the big question, and we have absolutely no idea. It's really hard to tell. You know, as I see, there's also questions about this. You know, of course, it could be consensual. We don't know whether it happened that, you know, like in the case of Denisova 11, the hybrid, you know, whether the Neanderthal mother lived with the Denisovan group, or whether the Denisovan father lived with the Neanderthal group, or whether, you know, the Denisovan father and the Neanderthal mother met at some point in the forest accidentally, and whatever happened, you know, because they were both in the mood, and, you know, neither of them wanted to sleep with their cousins, which was more or less the only option otherwise. It's hard to tell. You know, the cave is dark, and we can't see what was going on. What is clear is that it happened every time these populations interacted, and, you know, and this is common in history too. I mean, we know that any kind of, you know, colonization or even just larger migrations always were always linked to interbreeding between these populations. So it's not really surprising, but of course how it exactly happened it cannot be easily told, and sadly I don't think we won't be able ever to tell it.
Thank you. Have there been any studies in the Altiplano of South America for the high altitude gene? Where might that have come from?
It's actually really interesting. High altitude adaptation is a fascinating topic, and what is great is that it happened differently in different. So there's 3 like major groups in the world that are high altitude adapted: populations in the Himalayas, mostly in Tibet and Nepal, populations in the Andes, and populations in the highlands of East Africa. And what is interesting is that physiologically, these adaptations all happened differently. They actually work with different genes, and they have not just different genes, but also different physiological mechanisms. So it's only in Tibet that this variant comes from Denisovans. The variants in Peru, in particular, where it's been looked at, were evolved locally and independently. So yeah.
Yeah, the next question. Right, I'm puzzled at the complete lack of Neanderthal DNA in modern Africans. Since over the centuries, there has always been migration around the Middle East and across the Mediterranean. How is it that none of the Neanderthal DNA in European and Asian populations made it into Africa? And I had a question about that as well.
Yeah, no, that is a very good question, and it's complicated. So there's several reasons. One of them is methodological, and honestly, could be in part at least an artifact. There's probably very, very small amounts of Neanderthal DNA in African populations from various back migrations into Africa. We think, though, that this is probably very small because, honestly, up until about 10,000 years ago, by far the largest population in the world was in Africa. If we look, let's say, 100,000 years ago, you probably had 1 million people in Africa. And Neanderthals were maybe across all of Eurasia, maybe 30,000 individuals, if at all. And even 20, 30,000 years ago, there likely were about 10 times as many people in Africa as outside. So there was probably much more gene flow out of Africa as into Africa. And And, in present-day North African populations, for example, who have a considerable amount of Near Eastern and European ancestry, you see little pieces of Neanderthal DNA. The other thing is, though, is also that in these methods, we always have to use one group as the outgroup. And so usually the group that is used are Khoisan. And the assumption is simply that the Khoisan at the very tip of South Africa likely don't carry any Neanderthal DNA. So we kind of use them as the baseline, as zero. So what we always see is how much more Neanderthal DNA is there than in Khoisan. And so, 3% means whatever 3% more than in them. So if they have whatever 0.1%, then in reality, it's 3.1%. If you compare it directly, though, you can see that there's probably very little in most African populations, at least in South and in West Africa.
Thank you. Do you have a book underway or books?
No, not really.
How come?
I don't know. Haven't gotten around to it yet.
Or any publications you might?
Yes, many publications.
Point people to,
I have some popular ones. I wrote a popular one for a popular book last year. I can send that to you if you want to put a link up. There's also
and you can put that in the chat. And this research the original research was published in Nature. Is that right?
Some of it, yes.
A
lot of it. We had some in Nature, in PNAS, in Science, in the Journal of Human Evolution, in lots of different places. And
by the way, are any of these pieces available at the ROM or in Toronto or in Canada?
No, it's the ROM doesn't have an exhibition on human evolution, which is something that's been frustrating me since I moved to Canada. I tried to, we talked several times with some people who work there. The issue is that they need a major funder, someone who'd give the ROM, whatever, $500,000, $1 million or something, to actually create a human evolution exhibit. And they don't have a donor for that at the moment. Mm-hmm. If there were a donor, if any of you know someone.
If any of you would like to donate, yeah.
It would be great to have more on that. The closest I think to here is probably the American Museum of Natural History has.
Which is amazing, and not that far away. Yeah.
So I sent them casts of all this material.
Do you know where blue eyes came from?
Again, there's lots of different things. There's actually a couple of papers that say that some Neanderthals at least might have had blue eyes. I'm not completely convinced. Blue eyes likely arose they arose a long time ago. So we actually have the earliest individuals that clearly have blue eyes come from Europe, are about 12,000 years old. There's a Cheddar man from how's it called? Goes in the Cheddar Gorges in the UK. Anyway, a 12,000-year-old individual. And what is interesting about him is that he seems to have had very dark skin, present-day African populations, but blonde hair and blue eyes, which is kind of a combination that we don't see today. It seems that a lot of these combinations of pigmentation that we see today are actually quite recent. These are changes that the light skin in Europe only became really big about 5,000 or 6,000 years ago. Up until then, you had many individuals who were quite or even very dark skinned.
Thanks for that. What about the pygmy populations of? I'm not going to get that right. Right, that's right.
Yeah, I mean, Floresiensis is fascinating. The problem with Floresiensis is we only know them from a single site still. From the Denisova Cave. They haven't been discovered anywhere else really. And we don't have any DNA from them. It would be fascinating to know if they interacted with modern humans. But due to the high temperatures in Flores, so DNA preservation only works well in DNA preservation only works well if it's relatively cold and dry. So Siberia, is great for that. Parts of Europe also work. The Near East, Africa, Southeast Asia, not so good. And so we don't have any DNA from there. I'd love to have Floresiensis DNA, and I know my colleagues in Leipzig have tried many, many times. Matthias Meyer, who's responsible for developing a lot of the methods to extract DNA, every couple of years, he goes to Flores, gets some more animal bones, and plays around with them and tries to figure out. From one elephant bone, they actually got a little bit of DNA and could show that it was it's a pygmy elephant. But sadly, nothing from the human remains yet.
Thank you. So just time check, it's 4 o'clock. So if it's okay with you, let's take 2 more questions.
Yeah, I can do 2 more.
Would Denisovan and Neanderthal members recognize their differences? I guess this is going back to a question.
I think that's a good, I've always asked myself, you know, to me, the fact that both modern humans, I mean, that modern humans bred with Neanderthals and Denisovans, and also that Denisovans and Neanderthals interbred, indicates to me that they clearly recognize each other as potential mating partners. I think that, you know, they probably thought about each other as, you know, the kind of the ugly guys behind the mountains, kind of thing, you know, and maybe some were into them, you know, some people like the rugged, robust look. I don't know. But clearly they recognize each other as humans. So, you know, I do like it that we clearly all felt as one humanity, at least in, you know, in the most important ways.
What sort of insight might there have been among these groups regarding breeding with close relatives?
You know, we know from, you know, from ethnography, we know that usually there's strong taboos against breeding with close relatives. I think that sometimes, though, when the population is too small, there just is no other choice. And that's what it might have come down to it in these groups. You also have to imagine, of course, you know, probably the whole link between sex and conception and birth. We don't know how clearly that was understood. You know, we know that even in we just don't know how much they knew. I would imagine that, you know, ideally they probably would have preferred not to mate with their close relatives. But if your group really is only 20 individuals, you know, from baby to grandpa, that means that, you know, there's 4 individuals roughly who are roughly your age. There's not a lot of people to choose from. So yeah, it might have just come down to that. You know, you could either breed with a close relative or not at all.
Thank you. Well, this has been wonderful. Thank you so much for your amazing presentation. Such a cool story, and for, you know, taking us on a journey, Indiana Jones style. Thank you to our audiences for attending. Just a couple of things I would like to mention, as always, as we wrap up. If you have a moment to share your thoughts about the webinar or suggest a topic or a speaker, please fill out the survey. You will find the link in Zoom's follow-up email. This presentation was by popular demand, suggested by one of you. And I'd also like to mention that we are supported by memberships and donations. So if you'd like to become a member or contribute to our educational programs, including the webinars, you can do so on the website. And finally, join us for our next webinar on humanist chaplaincy, which is part of our chaplaincy webinar series next month on April 23rd at 3 p.m. Eastern, with Anthony Cruz Pantojas, who is a humanist chaplain at Tufts University. And as always, you can get more information about our webinars and register for them on our website or via our social media platforms and newsletter. Thanks again for attending, and Bence, thanks so much for your time.
Yeah, thanks a lot. It was fun.